Now, phytohormones, unlike microbial fermentations, plant cells do need, untransformed plant cells do need hormones in the medium. So, although the concentrations will range somewhere around milligrams per liter, but they are the expensive components, the microelements. So, you have to, apart from the microelements, so you must take care of the hormones, because especially with the untransformed cultures, plant hormones will be required to grow the plant cells. So, there are various class of plant hormones, starting from auxins, cytokinins, gibberellins, abscisic acid. So there are many plant growth regulators which you must investigate and optimize to find the optimum concentration, the optimum type, because even in a single class, you'll have many different types of hormones. These are sometimes naturally synthesized, and sometimes synthetic hormones. Now, because there are large number of parameters to be optimized, how do you do that? In a media itself, there will be some 15, 16 components, including microelements. Then you have an additional hormones as well. You have vitamins. These are all important for plant cells. So this will all account to some 15, 16 components. How do you think you should optimize it? How would you go about it if you are in an industry? Be loud. You must test it at? How would you test it? That was the question. Obviously, the media optimization should be done at the shake flask level itself. You do not wait till the reactor level. Please give, because I don't think he's audible to others. Right. Even before optimization, what is required to be done? Before optimization. Optimization is you are now have selected the right parameter and you are now going to optimize its concentration.
First one, any media preparation.
No, media preparation is a prerequisite. Come again, someone else? No, I was talking about how to identify what to pick up for optimization. All 16. All 16 will take so much time, isn't it? There can be so many permutation and combinations. Every component can be varied in a range. So imagine the number of experiments which you can have in hand to do only media optimization. And I told you n number of steps to reach to the final plant cell based bioprocess. How would you go about it? Where do you think what I just mentioned some time back will be of use here? Absolutely. So what do you need? Modeling comes in the play, which will help you to reduce the number of experiments to do these optimizations, especially when the experiments are large in numbers. So, as he was mentioning about response surface, that these are nothing but design of experiments, statistically designed experiments. But in the end, they are giving you a mathematical model to play around with. They will fit your data and give you a polynomial equation. And these polynomial equations will have coefficients which will be assigned values based on the best fit. Now this polynomial equation is ready to do predictions. Then in silico, you keep changing the values of all these 16 and you see the impact on what will be your dependent parameter. What are you doing it for? You are doing it for growth. You are doing it for product content per unit biomass. So they become your dependent parameters, and rest of your 16 or whatever number of media components are your independent parameters. Now, this itself is a big task. So, clever thing would be to, even in these 16, to only take those which are important. So, how do you do that? You have another tools, again, mathematical model tools with certain assumptions which you can use, which will help you to first screen these components and get you only top 3 or 4. It is wise enough to play around with. You'll generally observe when you'll rank these 16s, the impact of the change of the top rankers on the dependent parameter vis-a-vis the impact of a similar change on the bottom ones would be large difference. So why should you get bothered about those who are with the same amount of change are not so significantly impacting the product? Why should I spend my time, energy, and resource in optimizing that factor? I can let it be as it is, but my focus should be only on those parameters where I'm making a small change but showing a big change in the response. So that is what we do with screening designs. It will help you to work around and rank and identify the most significant parameters to play around for optimization. So you pick up those top 3 or 4, and then you use response surface methodology. It is again a design of experiment tool for optimizing their concentration in a given range. I'll not get into the details because this is not a forum for that, but just to let you know that how you should systematically develop bioprocesses, and industry cannot wait for single factor optimizations. So you must try to think when you are designing your experiments that in minimum number of experiments, how can I get best output and the most reliable output? Okay. So like in any microorganism or living organisms, there are major media, nutrients, and components. So I'll not get into the details. I think you can do a cell study. You can read the slides and know what is the importance, how phosphate is used, why nitrogen is of importance, why carbon is of importance. So I'll skip these things for your self study. Now, what should be kept in mind? I just spoke about how these statistical optimization tools can be used to reduce the number of hit and trial and the number of experiments to reach to the optimum media composition. Now, coming to after you have, let's say you have optimized the media now, then you, which includes your hormones also and your major salts, micronutrients. So now that is done. The next step is talking about the yield enhancement strategies. Now, mostly the media components, your response which you would take is 2 dependent parameters. One was biomass and the other was product content. If it would have been a growth associated product, I could have taken only single response, which was my product titer, because it had both the biomass component as well as the product yield. Because it is growth associated, there's a good chance anything which is improving growth will also improve my product. But if it is a non-growth associated product where there is a good probability of inverse relationship, and especially with secondary metabolites, there is a good chance. You should always have 2 parameters when you're optimizing anything. One is what impact that parameter can have on the biomass, and what impact does it have on the product content. Now, when I talk about precursor and elicitors, these are specifically product yield enhancement strategies. When I say product yield enhancement strategies, it would mean that they are targeted towards improving product content, amount of product present per unit biomass. Now, because I do not know how they are going to impact the biomass, although directly I know intuitively they are part of the biosynthetic pathway, so they are going to impact the product synthesis. But because primary is connected with secondary metabolism, I should be bothered about how is this increase going to impact the carbon flux towards my biomass. So it is important to have them separately when you are optimizing precursor and elicitor. So precursor is generally a compound which is an intermediate in or at the beginning of the secondary metabolite biosynthetic route, and therefore stands a good chance for product yield improvement. Now it can be endogenous, exogenous. Now saying that these are obviously intermediates, what am I trying to do? I'm trying to add them exogenously in the medium. So what am I assuming? What am I assuming? First step, absolutely. I'm assuming that if I give them outside in the cell will readily take it up and take it to the site of the biosynthesis where that product, because it is compartmentalized. So the what all things I'm assuming? First thing, exogenously it should be non-toxic. I don't care. It likes inside, so it will be liking outside as well. I'll add as much as possible and the cell will readily take it. the cell will very happily carry it to the compartment without getting degraded or metabolized, and there it will get utilized in the final reaction, and I'll see a good jump of my product yield. But that's not necessary. You do not know what is the nature of that intermediate. Some of the intermediates might be toxic to the cell. They might not be even available to the cell to be taken up. It has to cross a number of membranes. So that has to, so it has the availability of the precursor to come in is one prerequisite. The second is what?
Stability inside.
Absolutely. The rate at which it's getting utilized, the rate at which it is getting metabolized or degraded. So natural intermediate is a compound formed by the organism independent from the investigated biosynthetic pathway. Obligatory intermediate, a member of the path. Sometimes in this terminology, we also call them as indirect precursors. Now, as I told you, when Professor Nathalie was mentioning, it's a wide range of network in the secondary metabolism. Now, there is a good possibility if you are looking at an upstream precursor, there is a good possibility, although you have added, but what's the guarantee that it will go only in this branch where you want? It may go to some other branch. You have no control over it. So, you need to ensure that the purpose for which you are adding, it serves the purpose to go in the right branch. So, how can you ensure that? Any guess? I'm not asking to do any tweaking there. No genetic transformation. This is not a genetic transformation. Delivery mechanism means, again, you want to tweak genetically? So, generally, you use conjugates, which can easily, like cyclodextrins. If suppose you find it as toxic, you will try to club it in a mycelium-like structure so that it can be taken up by the cell membrane and then release it intracellularly. I was asking something else. How would you ensure that it is preferentially going to be, what can you do to ensure to be utilized in your preferred branch?
Giving the specific space to that of particular
No, there were so many secondary metabolites, and most of them, this is an upstream precursor. So if you are going to give a stress, there's a good chance that everything is going to get improved.
Even such that in order to counter that spread, that secondary metabolite, what other thing will not well.
So which means you need to know a specific stress which can lead to only that particular secondary metabolite. It's very challenging. Alpha tocopherol will have isomers alpha, beta, gamma, delta, alpha, and then tocopherol and tococriminol. So all of them will come as tococriminals. It's very challenging. What else you can do which is relatively doable? I was talking about something, a Q, indirect precursor. You know that something is getting diversified to various other branches. You are working on the top precursor. So there can be a chance where you can think of blocking the flow of this precursor to this branch without using down regulation. What can be done without using down regulation or genetic intervention? Adding inhibitors, ma'am. Absolutely. So those can be your, now not inhibitors, rather they are part of the biosynthetic pathway. But like, let me give you an example of azadirachtin. Azadirachtin is a secondary metabolite. The key precursor molecule is squalene. Squalene will also go towards sterol biosynthesis. The squalene will also go towards limonoids, where azadirachtin is also one of the limonoids. Now, what people have done is they have found that if they add cholesterol in the medium, there is a feedback inhibition. Cholesterol is a sterol which is formed. So if you are providing a lot of cholesterol, it will inhibit that path. What will happen? There will be presumably lot of accumulation for squalene. And then that squalene will preferentially flow towards limonoid synthesis and thereby improving azadirachtin production. So that was one of the examples of indirect precursors. Now, failure of these strategies can be because of a number of reasons. As I was mentioning, can be a lack of uptake, can be precipitation, can be diversion into alternative pathways, or lack of one of the enzymes, one or more of the enzymes in the bioproduction. Right now, we are assuming that the enzymes are all in place and their Vmax is still in place. They are not running at their Vmax. So the amount of enzyme which is required is already at its maximum. So then what? Then it is whether you keep pushing, you won't be able to see the result. So there are multiple factors which should be kept in mind for this strategy to be successful. Two methods of increasing precursor supply within the cell: by addition to the medium, in which case the uptake mechanism can be limiting; by selecting for resistance to precursor analogs, in which case the intracellular levels can be modified, like the pimelic acid I gave you an example. It's one of the precursors. This is just an example for the azadirachtin which I was talking about. So how to identify what can be the right precursors to be used? You try to have the information of the biosynthetic pathway which is available. You pick up the upstream ones or the downstream ones, and based on what can be readily taken up by the cell, and then so you do a screening of precursors, you optimize what can be the best precursor, and then optimize the concentration of that precursor to come to the best strategy. The second strategy I was talking about is cell permeability enhancer. Now, cell permeability enhancers, these will be agents which can reversibly permeabilize the cells. So, cell permeability enhancers are compounds which are not inhibitory to the cell growth when added, and at the same time have the ability to reversibly increase the pore size of the cell wall for better mass transfer. So, what all things can happen as a result of it? If there can be transient pores created, there can be better mass transfer of not only the product coming out, but also better mass transfer of the nutrients to and fro. So you may observe that to some extent, this will be helpful in the product leaching out of the cell and increasing better mass transfer in the cellular system. There are some examples where people have used different strategies for permeabilization. People have used organic solvents, but of course, the concentration is important. The type of organic solvent which you use is going to play a role. Coming to the most promising strategies until date, which can lead to maximum yield enhancements in plant cells, which is elicitors. As I was mentioning, elicitors are defined as molecules which generally stimulate defense or stress related responses in plants, which results in improved biosynthesis. So, from where are you going to get the cues? Again, what I was telling in my first lecture, you take the cues from nature. Why was this molecule produced in the plant? What is causing this molecule to be produced in the plant? If this is getting it is if it was targeted towards a particular fungal pathogen, what you can do is that you can make use of the cellular components of that fungal pathogen and use them in a dead state or in a live state and add them exogenously in the medium so as to give the signal to the cells and accelerate its secondary metabolism and thereby result in better product yields. Exogenous addition of elicitor molecules can be of both nature, biotic and abiotic. It is not necessary that mostly what happens, you must have, I don't know whether you remember, maybe you can go back and check. Professor Nathalie in her one of the first slides was showing a circle where different stress inducers, where UV radiation was mentioned, where light was mentioned, where salinity was mentioned, and fungal pathogens. Biotic were fungal, can be herbivory or anything. So sometimes they may not be very exclusive to a particular stress. So what can be exploited is you might be able to see a similar response if you use abiotic stresses or another type of biotic stresses, which may be easier to implement in the lab. So, for example, you may observe product elicitation or enhancement happening if you are going to use some salts, high concentrations of heavy metals, or if you want to use ultrasonication or heat shock. If any kind of environmental stresses you give, you may observe an acceleration in the secondary metabolism. Now, most of them are guided through calcium ions used as a second messenger. So, this was just a mechanism of action of elicitors. How do they work? There can be variations in membrane integrities or protein and phosphate metabolism, ethylene production, peroxidase activity. So, it's a signal cascade which runs through, which also involves your signaling molecules. If you remember what I was talking about, the involvement of jasmonic acid and salicylic acid in immune response or defense, there so people have also used signaling molecules addition in the medium because they are part of the signal cascade towards secondary metabolism enhancement. People have used them as elicitors and seen mostly a promising result impact on their secondary metabolite enhancement because these are generalized. They become because a part of the signal cascade. It does not matter what has been the origin trigger, but because they are part of the signal cascade, these can be used as, and they are pure molecules rather than using biotic elicitors, which will be complex molecules, which can bring with them some toxin as well, which can be a problem in the downstream. So if you are using extracts, so it is good to use single molecules. Now, what information which you got in the previous classes can be used to identify the right elicitors? Anybody? Okay. No, I was mainly wanting to hear about the lectures which were taken on the biosynthetic pathway.
Some of
That is going too deeper. To investigate will be another line of.
Ma'am, uh, ma'am. So we can look at the pathway. So if we are looking for a terpenoid compound, we can look at MVA and MEP pathways, how they can be enhanced. So the precursors can be taken from those pathways.
Sure. So that's also very good clue. Now you'll generally observe what are those rate limiting steps in the plant secondary metabolism. If you have a fair idea on the biosynthetic pathway, you can also have an idea of what enzymes are involved. Generally, like she was talking about few important classes of enzymes in the secondary metabolite biosynthesis. So many of these elicitors have been observed to change the expression level of these enzymes or activity of these enzymes. So generally, if you want to investigate in literature, you should be able to get the cues that what are the inhibitors for these enzymes, what are the inducers for these enzymes, or what all, like she was talking about the upstream pathways. It's a fair idea to look at the MEP and the MVA pathway. If not in this particular plant species, but in some other plant species, what has been reported to induce the expression or the activity of those enzymes, which can also be tried in the other systems. Okay, this is about the mechanism of action. Yeah, so what all things should be kept in mind when we are optimizing elicitor additions? Elicitor concentration, elicitor specificity, duration of elicitor contact. It's important. Why? Why do you think duration of elicitor contact is important? When should it be added and for how long should it be added? Very good. So, because these may have a detrimental impact on the biomass, the growth of the cell, so it's fair to look into when should you add it and for how long should you add it, the exposure to the cells. So, generally, when would you like to add it? How will you find out? what information would you be needing, which you would have collected during your shake flask? Why kinetics is of importance? Why should you be looking at when is the maximum biomass produced and then add the elicitor? How would that help your product yield? That you know anyways, that will be your end of the log phase. More biomass, the maximum biomass is the end of the log phase, isn't it? Right or wrong? So elicitor is, what is the mode of action of the elicitor? What is it impacting on? It is impacting your product formation rate, isn't it? Product yield per unit biomass. And from your kinetics, what data did you get? Product, substrate, biomass. So what can you get? Specific product yield as well, isn't it? Specific product formation rate that can be obtained, which is nothing but the yield with respect to how is it changing? Gram of the product per unit biomass per unit time. So now if you know specific product formation rate, when is it maximum? Ideally, when should you add it? Because now this is the point where all the precursors, everything is maximally getting utilized for product formation. Before that. So whenever you are observing your product yield is maximum, you assume that the product formation was happening near that point before you could see that in the medium on the cell. So you try to work around that point 48 hours before that, 24 hours before that, 72 hours before that point, and see what impact. So this is how they manage the addition of elicitors. Similarly for precursors, because it is the point where their maximum utilization of precursors towards your product is happening, possibly. So it is logical to use that point of addition, because then there is a good chance that when you add, it will be preferentially used in your preferred branch of the secondary metabolite. Elicitation in cell suspension cultures can result in different products, may show high levels accumulation at different time points, because it depends on your elicitation strategies. Generally, these are stress inducers. So under stress environment, the plant will be gearing up the cell will be gearing up its entire secondary metabolism. So you might be able to observe other products also coming up. Now, coming to types of elicitors, I did mention biotic elicitors. Biotic elicitors are generally directly released by the microorganisms and recognized by the plant cell. They can be enzymes, they can be cell wall fragments. They will be formed by the action of microorganisms on plant cell walls, wherein the fragments of the plant cell wall can also be an indicator to plant cell that something bad has happened. Formed by the action of plant enzymes on microbial cell walls. So wherein chitosan, glucans, all these can also act as biotic elicitors. Abiotic elicitors, any kind of stress inducers have been reported as abiotic elicitors. For example, people have used UV light, denatured proteins, freeze-thaw cycles, or agros, heavy metals. But please keep in mind when you are using abiotic elicitors, if you are using very toxic chemicals like heavy metals, that can be a problem in your downstream processing, especially when you are working with pharma components. So one has to be judicious enough in choosing what kind of elicitors are you using in your system. Chemicals with membrane destroying ability, you can, although all this in R&D, you can do anything. When you're doing research, you're free to do anything. But when you're developing a technology for commercialization, you have to be careful because safety is a consideration in the final product. The downstream processing, everything is going to be an added cost to remove all those components. Okay, so biotic elicitors can have n number of components or cellular components, can include polysaccharides, lipids, or oligosaccharides. They can also act as so endogenous elicitors. So there are various classification in elicitors themselves. You can class them as biotic, abiotic. You can class them as endogenous, exogenous. Now, endogenous elicitors are of plant origin and are generated by the interaction between the pathogen and the plant. The exogenous elicitors will be which will be originating in the invading organisms. Abiotic elicitors, I've already told you, they will be abiotic in nature. So one of the other strategies to improve productivity is also immobilization of plant cells. The use of high biomass levels. So in order to achieve high productivities, as you people were also saying, high biomass concentration is required. So you can take the advantage of immobilization to have these high cell densities in smaller volumes. So the plant cell immobilization is an opportunity where large amount of cells which are active can be immobilized, giving you high cell densities for only production to happen continuously, thereby increasing the productivity. Now, immobilization of plant cells can be used for various purposes, especially it can be used for biotransformation. Majorly, you will see in literature that people are using organ cultures, which are called as hairy root cultures. For they immobilize the hairy root cultures and they use them for these biotransformation reactions. Biotransformation means that conversion of a particular substrate to your desired product by using plant cells or organ cultures as repository of enzymes. So a low cost substrate is getting converted to a high cost product. Synthesis from the precursor. You can also add the precursor again because you are using the whole cell as the enzyme repository. So there is a good chance that the precursor can be converted to product. De novo synthesis of compounds, of course, what we have been talking about in plant cell based bioprocesses. Due to its inherent ability to produce these phytochemicals when they are growing, so you allow the cells to grow by providing carbon source, the media nutrients, and during their growth, they are also producing your product. So starting from scratch. Now the separation, the advantage of immobilization, whether it is by microbial cell immobilization or plant cell immobilization, the advantage is that it can separate the cell from the medium and therefore the downstream processing cost is reduced. Now the catch there I was mentioning was that the product has to come out in the medium. Most of these secondary metabolites are intracellular. So the strategy which we had earlier studied about, which was cell permeability enhancement, that is optimized and added so as to have the product leached out. And the added advantage to this is that because of the product leaching out, it acts like a push towards product synthesis because it acts like source and sink. So if the product is coming out, so the rate of forward reaction is driven. So that may also lead to improved product synthesis intracellularly. Now, because these are immobilized systems, in comparison to free cell systems, the problem of cell aggregation can be avoided, which can be a problem because of the inherent property of cells to come together. So the cell aggregation and causing problems in mass transfer. So it's a completely rather different kind of cultivation. There is no direct comparison between free because free cell suspensions are completely different and immobilized systems, their reactor designs are completely different. Sometimes what happens, because plant cells are relatively shear sensitive, immobilizing them is also an advantage to keep them away from the shear effects of the hydrodynamics present in the reactor systems. Okay, the efficiency of this to be a successful strategy lies in the fact that the rate of release of product is what is your rate limiting step rather than rate of synthesis. So the rate at which the product will be released from the immobilized cells becomes your rate limiting step for productivities to be achieved, high productivities to be achieved. It may work both ways. Sometimes what happens, the immobilization, the microenvironment which gets created inside can be an elicitor acting phenomena to have further acceleration in secondary metabolism. The prerequisites for successful immobilization of plant cells strategy would be non-growing cells must produce products. There's a good chance because these are secondary metabolites, specialized metabolites. Products must be released from the cell in the medium, as I mentioned. Now, we know that they are relatively shear sensitive. There is also a need for cell to cell contact. I was mentioning that sometimes you will observe in the lab that the finer is the cell suspension, the product yield is compromised. So sometimes we do try to differentiate and make it into a two-stage cultivation process, where the first stage you try to improve the biomass productivities are maximized, and then in the second stage you implement conditions which can be stress enhancing conditions, or trying to club them together, or even to bring in differentiation in these cells such that the product synthesis is maximized. Because secondary metabolite synthesis can also be a function of cell differentiation. There are different ways in which you can immobilize. I'm sure those who have done fermentation courses, they know how cell immobilization can be done. So this is again a topic which for your self-study, the information is available, where different techniques can be used for cell immobilization, ranging from covalent coupling, natural affinity, which will include adsorption, addition, agglutination, or cross-linking by using polyfunctional agents, or using temperature to change the consistency, the physical properties, like when you use agarose gels. Or you also use entrapment like calcium alginate beads. Now, what care should be taken while selecting the polymers for immobilization? They have to be inert, non-toxic to the cells, and they have to be economical in nature, and obviously they have to be available to meet the demand. And they should also, the process of immobilization should not be such that it can diminish the enzyme activity of the cell. Okay, so I'll not get into the details of the process of immobilization. Possibly you can go through yourself how gel entrapment is done, because these techniques are common to microbial fermentations or enzyme immobilizations. All those techniques which are used for immobilizing cells can also be used for microbial cells, can also be used for plant cells also. Now, coming to the reactors which can be used for immobilized cells, how they are different from stirred tank reactors. They are generally packed columns. So, these packed cell reactors, there's a limitation of large amount of pressure drop, because how to improve the mass transfer requirements in these? The improvement in mass transfer requirements is done by converting these packed bed columns to fluidized bed columns. Now, fluidized bed column, this is again a technique which is common to both immobilized microbial systems or plant cell systems. The basic difference is that in the fluidized bed systems, you make use either counter current or same direction of the currents. You make use of the gases is to mix with the liquid, the substrate stream, such that there can be recirculation happening, which can cause better mixing. Okay. Now, once it has been immobilized, how do you take care of the cell viability? How do you know that the cell is intact to continue to give you the product? So there should be techniques by which you can check the viability of the cells. Especially in immobilized systems, you can do that either using dissolved oxygen variations in the bulk medium, or you can find out using certain dyes which can give you an indication for the viable cells. Okay, I was talking about the packed bed reactors. Then you have the fluidized bed reactors. Large gas volumes will help you to have to facilitate low liquid circulation rates. Because what happens if you want to improve the mass transfer and if you try to increase the liquid circulation rates, because of the increased turbulence inside, there is a good chance of shear rates, and wherein they can rupture, the cell viability can be compromised. So then you make use of the gases as well, so that with a lower liquid recirculation rate, you can afford better mass transfers because gas is also helping in mixing. Now, there are membrane bioreactors, although they are not very well known for plant cell systems. They are very well known for mammalian cell cultures, hollow fiber reactors. One of the reasons is that plant cells are bigger in size and they tend to aggregate. So many a times, because these are long drawn fermentations, many a times this will clog the membranes. So although these are reported, you'll see one or two such reports there, but they are not very frequently used reactor systems. But nonetheless, these are some of the membrane bioreactors which can be used for immobilizing cells. Now, how do we immobilize cells? Like, for example, in organ cultures, when we are working with hairy roots, they are self-immobilizing in nature. They need support and then they will come together for further branching to happen. So, an inert material is used to inoculate these roots in those inert materials before they can begin, the inoculum can begin regrowth and branch it out, and the biomass densities can be improved. Okay, so this brings to an end